Ever wonder how a paper cut just heals like magic, right? Well, that's one type of cell division, but then there's the process that created you in the first place, and that's a totally different ballgame. You see, our bodies have these two different playbooks for making new cells, and getting the difference between them is honestly key to understanding life itself. So let's dive into mitosis versus meiosis. Okay, so why bother with two systems? I mean, why does your body need a perfect photocopier for some jobs and a kind of wild genetic remixer for others? Well, the answer really boils down to their two totally different missions. One is all about keeping things stable and consistent, and the other, it's all about shaking things up and creating variety. All right, first step, mitosis. And the best way to think about this is like the body's super precise, high-tech photocopier. Its whole job isn't to be creative or make something new. Nope, it's just to make perfect, identical copies of cells that are already there. This is the workhorse process that lets you grow, replaces your old, worn-out cells, and heals up those cuts. So mitosis is happening all the time in what scientists call our somatic cells. And really, that's just a fancy word for all the regular cells that make up your body. You know, your skin, your bones, your muscles, pretty much everything except for the special cells used for reproduction. Those are called germ cells, and they are set aside for a completely different process. And this whole process, it's like this elegant four-step dance. First, you've got prophase, where the DNA scrunches up into these visible chromosomes. Then, in metaphase, they all line up perfectly right down the middle. Super orderly. Then comes the main event, anaphase, where identical copies get yanked to opposite sides of the cell. And finally, in telophase, this cell pinches in the middle and divides. Boom, one clean split. And what's the end result? It's simple, really. You start with one parent cell that has a full set of 46 chromosomes. We call that a diploid cell. And you end up with two daughter cells that are just perfect genetic clones of the original. Each one has its own complete set of 46 chromosomes. Just perfect copies, every single time. Okay, let's switch gears and talk about the other side of the coin, meiosis. Now, if mitosis is that perfect photocopier, meiosis is like a genetic DJ, a remix artist. Its one and only job is to create sperm and egg cells. And its goal? It's the exact opposite of mitosis. It's not about making perfect copies. It's about intentionally creating genetic variety. Meiosis is really a two-act play. And in the first division, meiosis I, something absolutely incredible happens right out of the gate. During prophase I, the chromosomes you got from your mom actually pair up with the ones from your dad, and they physically swap pieces of DNA. This is called crossing over, and it creates brand new combinations of genes that have never existed before. Then, when the chromosomes get pulled apart, it's the pairs that separate. This is huge because it means that after just one division, the new cells already have half the original number of chromosomes. Then comes the second act, meiosis II. And this part is going to look really familiar because it's almost exactly like a regular round of mitosis. The chromosomes line up in the middle, they get pulled apart, and the cells divide. The key difference? The cells doing this dance are already working with just half the genetic material they started with. And so, what's the final result of all this? Well, instead of two identical cells like in mitosis, meiosis gives you four genetically unique cells. Each one is called haploid, which just means it has a single set of 23 chromosomes. They're now ready to hopefully combine with another gamete during fertilization. And the most important part? No two of them are exactly alike. So let's just put it all side by side for a second. The differences are pretty stark, right? Mitosis is for growth and repair, while meiosis is strictly for sexual reproduction. Mitosis involves one division and you get two identical cells. Meiosis takes two divisions and you end up with four unique cells. It's really the difference between cloning and creating. Okay, so we know meiosis creates these unique cells, but how does it do it so well? And maybe more importantly, why does nature even bother with all this extra work? This is where meiosis really shows off its evolutionary genius. It basically has two main tricks up its sleeve for shuffling the genetic deck. And that genetic shuffling, it's not just some neat little trick, it's the whole point. You see, this variation is the raw material for evolution. It's what gives a species the flexibility to adapt to, new diseases, changing climates, you name it. Without that variety, a species is incredibly vulnerable. So let's look closer at that first mechanism, crossing over. This is literally where the chromosomes you inherited from your mom and dad physically break and swap little segments of DNA. What you get at the end is a chromosome that's a completely new mosaic,
a patchwork quilt of your parents' genes.
The second trick is called independent assortment. So when those chromosome pairs line up in the middle of the cell, how they orient themselves, which one's on the left, which one's on the right, is completely random. It's basically a coin flip for each of the 23 pairs. Now that might sound simple, but think about this. That process alone creates over 8 million possible combinations of chromosomes in the final sperm or egg. And that's before you even add crossing over into the mix. It's just staggering. Now, these processes are incredibly precise and usually run like a well-oiled machine. But they're not perfect. They're not infallible. And when cell division messes up, the consequences can be incredibly significant for a person's health. The most common type of error is something called non-disjunction. And you can think of it as a failure in quality control. The machinery that's supposed to pull the chromosomes apart just slips up. What happens is one daughter cell gets an extra chromosome, while the other one is left one short. And this state, having the wrong number of chromosomes, is known as aneuploidy. Now, if this kind of error happens during mitosis out in the body somewhere, it can create these patches of cells that have the wrong chromosome count. This is actually a factor in some forms of cancer, where that genetic imbalance can be the very thing that triggers uncontrolled cell growth. But when non-disjunction happens during meiosis, the error gets baked right into the sperm or the egg itself. And that means every single cell of the resulting offspring will have that chromosomal abnormality. This is the origin of many well-known conditions, like Down syndrome, which is caused by an extra copy of chromosome 21, as well as others like Edwards, Pei-Tau, Klinefelter, and Turner syndromes. So it seems simple enough, right? Meiosis is all about creating genetic diversity. That's what we've been saying. It seems totally obvious. But what if that wasn't its original purpose at all? The evolutionary story of meiosis has this really fascinating twist, and its origin might not be what you think. Yeah, so we've all been taught that meiosis is nature's great genetic shuffler, and that this diversity is its main benefit. It just feels right, doesn't it? But some evolutionary biologists have come up with a different and honestly a pretty surprising idea about its original purpose. Get this idea completely turns the common wisdom on its head. It suggests that the first job of meiosis wasn't to encourage genetic mixing, but to actually control it, to put a leash on a process that could otherwise be really, really dangerous. So here's the theory. As early life forms started getting bigger, more complex genomes, they filled up with a lot of repetitive DNA. Now, the ancient process of recombination could get confused by all this and try to swap pieces between the wrong chromosomes, which would cause catastrophic mutations. So meiosis came along as the solution. By forcing the matching chromosomes from each parent to pair up perfectly along their entire length, a process called synapsis, it basically guaranteed that any DNA swapping only happened between the correct partners. It was the safety check, first and foremost. So it looks like meiosis probably started as a safety mechanism, and only later did evolution repurpose that feature into the incredible engine of diversity we see today. It's just a powerful reminder that even life's most fundamental processes have these hidden, surprising stories. It really makes you wonder, doesn't it? What other secrets of survival are hiding in plain sight, written in the code of our own DNA?